Assalamu alaikum. We'll continue with the histology of the urinary system. In this lecture, we'll discuss the histological features, both the light and the electron microscopic appearance of the proximal tubule, the distal tubule, collecting tubules and ducts, juxtaglomerular apparatus, the ureter, urinary bladder, and the male and the female urethra. This diagram showing you the different parts of the nephron. As you know, the nephron is the functional and the structural unit of the kidneys. The nephron starts from the renal corpuscle, which we have already discussed. Then there is a proximal convoluted tubule. This proximal convoluted tubule descends to the descending limb of the loop of the Henle. Then it forms a U-shaped segment, and it has a thin segment and a thick segment, thick ascending segment, and then ascending limb. It joins the distal tubule. The distal tubule has also the straight portion and the convoluted portion. Then distal tubule connects with the collecting system, that is the collecting tubule, and the collecting tubules that join the collecting ducts, which are poured through the papillary duct into the minor calyces. So we'll discuss the histological features of each part of the nephron one by one. The proximal convoluted tubule is a very tortuous tubule, and it is longer than the distal convoluted tubule. So in the sections of the cortex, there are more proximal convoluted tubules are more visible. So the cells of the proximal tubule, they reabsorb 60 to 65% of the water, and which is filtered in the renal corpuscle, along with almost all of the nutrients, ions, vitamins, and small plasma proteins. The water and its solutes are transferred directly across the tubular wall and immediately taken up by the peritubular capillaries. This diagram shows you the section of the epithelium of the proximal and the distal tubules. The proximal tubule has the brush border and eosinophilic cuboidal epithelium, as compared to the distal tubule, which has the sparse short microvilli, and it is less eosinophilic. In this photomicrograph, you can identify a renal corpuscle. And now you can see at the tubular pole, the parietal layer of the Bowman's capsule, which is the simple squamous epithelium. It continues with the cuboidal epithelium of the proximal tubule. Now this cuboidal epithelium, you can see that the cells, they are eosinophilic, and the nuclei, they are the rounded and central in position. The proximal tubule has a brush border, which appears in the light microscopic appearance or the routine H&D stains. It appears as a fuzz border. The cells of the proximal tubule, they have the numerous mitochondria, therefore it appears more acidophilic. So the capillaries and the other microvasculature components, they are abundant in the surrounding connective tissue. The cells, they are large. Each transverse section of the proximal tubule typically contains 3 to 5 rounded nuclei. The ultrastructure of the proximal convoluted tubule reveals its all important features of the cuboidal epithelium. It has the long microvilli, which are they are very dense, and the abundant pinocytotic vesicles, they are present in between these microvilli. Now these small proteins, they are brought into these cells not specifically by the pinocytosis, and they are degraded in the lysosomes, and the amino acids released basally. The apical ends of the adjacent cells, they are joined by the zona occludens, whereas the basolateral surface, they are characterized by the long invaginating folds of the membrane. Now these folds, they are filled with the elongated numerous mitochondria. As you know that proximal convoluted tubule is involved in the active transport of the sodium and the glucose. So therefore, the mitochondria, they provides the energy for the active transport. So these folds, they also increase the surface area of these proximal convoluted tubule cells for the ion exchange. The proximal convoluted tubule, they continues as a proximal straight tubule which enters the medulla and becomes the nephron loop. And this nephron loop is U-shaped structure with a descending limb and an ascending limb. So both of these limbs, they are composed of the simple squamous epithelium. Now the ascending thick limb and this thick limb, you can see here, it is more cuboidal near the cortex, whereas the thin limb, it is simple squamous epithelium. It has the simple squamous epithelium. So the lumen of this segment of the nephron is wide and the wall consists of the squamous epithelial cells whose nuclei, they protrude slightly into the lumen. Now, this photomicrograph showing you the segments of the loops of Henle, the proximal convoluted tubules. This is the thin segment, and this is the thick ascending limb of the loop of the Henle. This section showing you the collecting ducts, and also this is the tubules, and these are also the collecting tubules and the ducts. Now, the distal convoluted tubules, they have also composed of the simple cuboidal epithelium with the centrally placed rounded nucleus. But the distal convoluted tubules, they are less eosinophilic and they have sparse microvilli. Therefore, the brush border is not prominent in the distal convoluted tubules. In the transverse sections of the distal tubule, there are more nuclei. They can appear in the more nuclei as compared to the proximal tubule. So in the sections of the proximal tubule, only 3 to 5 nuclei of the cells they can be seen, whereas in the distal tubule there are more. The lumen of the distal tubules is wide as compared to the proximal, and it is clear as compared to the proximal, which appears fuzz-filled. The electron micrography appearance of the distal tubule shows the basal membrane enfoldings that shows that it is also involved in the transport of the ions, and it has also the mitochondria present in these basal enfoldings. The distal tubule, they secretes the hydrogen ions and the ammonia into the tubular urine, an activity which is essential for the maintenance of the acid-base balance in the blood. The cells of the distal tubule, which comes in contact with the arterioles at the tubular pole, they change their configurations. That is, they become thickened, and from cuboidal epithelium, they change their configurations to tall columnar cells. So these cells, they are called the macula densa cells. So the adjacent to the macula densa cells, the tunica intima, the tunica media of the afferent arteriole, it is also modified. So you can identify here, this is the tubular pole, and this is the distal tubule which comes in contact with the afferent and the efferent limbs, and the cells, they become columnar in shape as compared to the rest of the distal tubule. Now this is a structure showing you the juxtaglomerular apparatus. So the straight part of the distal tubule makes contact with the vascular pole of the renal corpuscles of its parent nephron, and it forms the macula densa cells, which are the tall columnar cells, whereas the rest of the cells, they are simple cuboidal cells. Then between the limbs, there are the extramesangial cells, which we had discussed in the previous lecture. Now the cells close to the afferent limb, afferent arteriole, the smooth muscle cells, they also change their configuration. And these extramesangial cells, they have the zymogenous granules. They are modified, and they are also called the Lesch's cells. So this setup, that is the macula densa cells, the extramesangial cells, and the Lesch's cell, this constitutes the juxtaglomerular apparatus. Now, this juxtaglomerular apparatus is responsible for the feedback mechanism that allows the autoregulation of the renal blood flow and keeps the rate of the glomerular filtration relatively constant. These Lesch's cells, they also transmit the signals from the macula densa into the glomerulus and affecting the vasoconstriction there. This photomicrograph showing you very clearly these macula densa cells and all the components of the juxtaglomerular apparatus. That is the macula densa cells, the JG cells, and these extramesangial cells, the Lesch's cells and the extramesangial cells. Now the collecting tubules and the ducts. This collecting tubules, they are the last part of each nephron, and they join to form the larger straight collecting ducts. They run into the tips of the medullary pyramids and empty into the minor calyces. So the collecting tubules, they are lined with the cuboidal epithelium. The cells, they are cells of the converging collecting ducts. They are more columnar. Along the entire extent, collecting tubules and the ducts, they are composed mainly of the weakly stained cells, which are called the principal cells. These principal cells, they have few organelles and scanty microvilli. These are the principal cells. So ultrastructurally, the principal cells, they can be seen. They have also the basal infoldings consistent with their role in the ion transport. And scattered among these principal cells, there are also darker intercalated cells. They have the abundant mitochondria, and they regulate the acid-base balance by secreting the hydrogen ions and absorbing the bicarbs. In the medulla, the collecting ducts, they are a major component of the urine concentrated mechanism. The cells of the collecting ducts are particularly rich in aquaporins also. These aquaporins, they are the integral proteins. They are found in the most of the cell membranes. They function as selective pores for the passage of the water molecules. So aquaporins, they are sequestered in membranous cytoplasmic vesicles. Now in this diagram or a sagittal section, you can see the renal papilla, and it shows all the numerous collecting ducts, which are also called the ducts of the Bellini. They are converging towards the tip of the pyramid, renal pyramid, that is the renal papilla, and from the renal papilla into the minor calyx. Now you can see here the mucosa of this calyx. This is the mucosa, or this is the minor calyx, and the mucosa of this calyx, they contains the dense connective tissue, which is stained blue, and adipose tissue. So the ducts, they are embedded in the interstitial tissue that also contains the thin limbs of the nephron loops, and this is a Mallory trichrome stain, which is a special stain. Now, in this enlarged area, you can see that the collecting ducts, they are these tubules, and this is the interstitium, stained blue. And this lining epithelium, which is the columnar epithelium, it's transition into the urothelium. The urothelium, which is a transitional epithelium. So the transitional epithelium, it lines the minor calyces, the pelvis, the ureter, and the urinary bladder up to the urethra. Therefore, it is also called the urothelium. This is a diagram showing you the cut section of the ureter. You can see here the ureter is in this transverse section of the ureter. It is the lumen is thrown into folds, and it's called have a star-shaped lumen. The mucosa consists of the epithelium and the lamina propria. The epithelium is a special type of the epithelium, which is the transitional epithelium or the urothelium. Then is the coat of the muscularis layer, and then is the adventitia. So the urine is transported by the ureters to the bladder, where it is stored until empty during micturition via the urethra. So the calyces, the renal pelvis, the ureters, and the bladder, they have the same histological appearance, with the walls becoming gradually thicker as they become close to the bladder. The mucosa of these organs is all lined by the transitional epithelium or the urothelium, and it is surrounded by a folded lamina propria and submucosa, followed by a dense sheath of interwoven smooth muscle layers and adventitia. So urine moves from the renal pelvis to the bladder by the peristaltic contractions of the ureter. Now, this is a diagram showing you the urothelium or the transitional epithelium. The urothelium is composed of three layers of the cells. This basal layer of the cells is consisting of the stem cells or the small basal cells, which are resting on this basement membrane. Then there are 2 to 3 layers or four intermediate region forming the several layers of the cuboidal to columnar cells, and then there are the superficial cells, which are the dome shaped cells, and they are called the umbrella cells. They are bulbous and they are projecting into the lumen. uh, they are occasionally multinucleated or binucleated, and they are highly differentiated to protect the underlying cells against the cytotoxic effects of the hypertonic urine. So they are joined together by the tight junctions, and they have also a glycocalyx coating over them. So they are specialized to hold the urine. Now, the characteristic feature of this urothelium or the transitional epithelium, as you have already studied in the first years, that they have a tendency to contract and relax. That is, they are stretchable, so they can be distended, and they can be in the full bladder. These cells, they become distended, and these dome-shaped cells, they become flattened. Now, this is a diagram showing you the urinary bladder. Now, urinary bladder, you can see here that it is lined by the transitional epithelium, the mucosa that have the transitional epithelium, and this is the lamina propria. Below is the thick layer of the muscles, which is called the detrusor muscle. This detrusor muscle is arranged in three layers, right? And this is inner layer, middle layer, and the outer layer. And then is the adventitia. You can see here the distended bladder, and this is an empty bladder. So when the bladder is empty, you can see the transitional epithelium. It becomes the dome-shaped cells. They are clearly visible, and the cells, the layers become more extensive, and they become tall columnar-like cells in the distended bladder. This is again a diagram showing you the layers of the urinary bladder. You can see here that this is the urothelium or the transitional epithelial lining in the mucosa. Below is the lamina propria, and there is a thin layer of the submucosa. Then the layer of the muscularis or the detrusor muscle, arranged in the three layers: the internal longitudinal, middle longitudinal, and the outer longitudinal layers. This is showing you the contracted state of the urinary bladder, in which you can clearly see the dome-shaped cells of the transitional epithelium. This is a special stains, and when the bladder is full, you can see that these dome-shaped cells, they become flattened, and these layers, they does not appear as they appears 2 to 3 layers, whereas in the the empty bladder, they appear 4 to 6 layers, and the surface cells, they become flattened in an distended bladder. The urethra, it is also aligned with the transitional epithelium or the urothelium. It is lumen is also folded in itself, and longitudinal folds, and it gives it a distinctive appearance in its cross section. but there is a difference in the male urethra and a female urethra. The female urethra is short and is a part of the urinary bladder also, but the male urethra is long and it has three parts. So the male urethra consists of the three segments, that is the prostatic urethra, the membranous urethra, and the spongy urethra. The prostatic urethra extends to the prostate gland and is lined by the urothelium, whereas the membranous urethra, it consists of a short segment and it passes through an external sphincter of the striated muscles, and it is lined by the stratified columnar epithelium or pseudostratified epithelium, whereas the spongy urethra is enclosed within the erectile tissue and is lined by the stratified columnar and pseudostratified columnar epithelium, with more distally, when the area of the skin, it is stratified squamous keratinized. The female urethra is exclusively a urinary organ, and in the female urethra, it is lined by the transitional epithelium, and then by stratified squamous epithelium in some areas, or pseudostratified columnar epithelium. The middle part of the female urethra is surrounded by the external striated muscle sphincter. So this is all about the histology of the urinary system, and in which we have discussed the proximal tubule, the distal tubule, the collecting tubules, and the ducts, and these histology of the urinary bladder, ureters, and the parts of the male urethra and the female urethra. So if you have any queries, then you can ask me on WhatsApp. So thank you so much.